Hello, thanks for being here today, and thanks Immcantation for the invitation in this meeting. In this talk, I will be presenting our work on Integrative Bioinformatics for characterizing atypical memory B cells through single-cell RNA-seq in COVID-19. And I would like to start talking about how is the B cell response in COVID-19. Once the virus enters the body and interacts with naive B cells, these cells can take two main pathways. The extrafollicular pathway that is like for a rapid antibody response. The naive cells get activated and went into the differentiation of short-lived plasma cells to produce antibodies, but with low affinity to the antigen. At the same time, naive B cells can be activated, migrate, and proliferate to form germinal centers. And this process can lead to the development of memory B cells and long-lived plasma cells to produce antibodies with higher affinity than in the extrafollicular pathway. Also, there have been reported atypical memory B cells in COVID-19 and other diseases, but the development pathway and the roles of these cells are still unclear. And this is the normal or the standard response of the B cells against the virus. But what happened with severe cases of COVID-19? Well, some studies focus on the B cell response have been observed that when they compare severe versus non-severe cases, patients with severe exhibited a strong extrafollicular response and neutralizing antibodies, which were insufficient to resolve the disease. Also, another study reported that in postmortem analysis of lymph nodes and spleens from COVID-19 patients showed an absence of germinal centers. So this study aligns pretty well with this other. And based on these results and aiming to better understand the B cell response, one of our main objectives was to perform an integrated bioinformatic analysis at the transcriptomic single-cell level to characterize B cell responses, including atypical memory B cell response in convalescent COVID-19 donor. And to address this, we followed this methodology. First, we select convalescent COVID-19 patients with different clinical manifestations. We collect blood samples from them, and then we isolate the B cells to perform the S1 and RBD specific B cell magnetic enrichment. Once we have these protein-specific B cells, we continue with the single-cell 10x barcoding, followed by BDA and gene expression library sequencing. When we obtain the sequences, we continue with the data analysis. For the data analysis, we follow this workflow. First, for the pre-processing, we use the Cell Ranger Multi pipeline from 10x Genomics to process the raw data into the necessary outputs to continue with the pre-processing part, which includes quality control, normalization, batch correction, feature selection, and the visualization, for example, UMAP or TSNE visualization. Next, in the downstream analysis, once we complete the pre-processing, first we start with the clustering and cell annotation. For the cell annotation, we use manually cell annotation with the expression of canonical markers to identify the different B cell subsets. And when we have the annotated cells, we continue with the enrichment analysis using gene ontology, biological process, and molecular signature databases. Also, we perform trajectory analysis with a slingshot, and we perform repertoire analysis with the Immcantation framework. Particularly, we use change-O to convert these BDJ cell ranger outputs into AIR standard format for the next analysis. For example, SHazaM to analysis of the somatic hypermutation rates, and ALAKASAM for the physicochemical properties of the BCR. And as part of the results, in the clustering and cell annotation, we process more than 18,000 cells, and we identify 10 different B cell subsets. For example, we identify in this part transitional B cells T1, T2, and T3. We also identify naive activated B cells, 5 different groups of memory B cells, including atypical memory B cells, which are characterized by the low or null expression of CD27 and CD21. And we identify as well antibody secreting cells. This is the distribution of cells across the conditions, and we observe that severe and moderate patients have the higher number of cells and also higher heterogeneity. And when we observe the frequency of the cells by condition, something that called ovation was the transitional B cells in critical and severe group were the higher with the transitional B cells. Critical, for example, in, well, T2, transitional 2 in critical, and transitional 1 in severe patients. This high frequency of transitional B cells may be linked to the antigenic persistence in severe and critical cases, and this for the development of mature B cells. But the fact that these cells remain in a transitional phase during convalescence suggests that these cells may contribute to the disease severity. And another thing that called rotation, that was atypical memory B cells, was higher in the severe group. In the differential express gene analysis across the cell type and condition, I'm showing here this heat map where in the top are colored the cell types and followed by condition, and the genes highlighted in red are the genes related to the proinflammatory response, and the blue are the related to activation and antiviral response. In this case, we observe that severe and critical cases, as can we see here, are exhibited upregulation of proinflammatory genes, whereas mild and moderate groups show higher expression of antiviral and activation genes. For the BCR repertoire analysis, the results show that the different rates of somatic hypermutation correlates with the cell differentiation, with the stage of cell differentiation. For example, transitional cells that are the more immature cells express less or null somatic hypermutation rates, but the more differentiated cells express or present more somatic hypermutation rate. And here we can we focus more on atypical memory B cells that show lower rates of somatic hypermutation in comparison with those switch memory B cells, but higher rates than non-switch memory B cells. We also perform analysis of physicochemical properties using SHazaM. And in this case, we didn't observe any statistical significant differences between atypical and conventional memory B cells. Well, for this comparison, we group all the memory B cells and exclude atypical memory B cells to compare each other. And we analyze the amino acid length, the hydrophobicity, polarity, and also the content of basic and acidic residues, but in this case, we didn't observe any difference. So it seems that this group of cells have the same physicochemical properties, but different somatic hypermutation rates. Also, when we analyze the gene usage in comparison of atypical versus conventional memory B cells, we observe, well, in red are the gene usage from atypical, and in blue, the gene usage from conventionals. In here, we observe some difference in gene usage between atypical and conventional. So at this point, we wanted to understand more the atypical memory B cells, and we wanted to know where was the possible development pathway of these cells in COVID-19. There are some studies that report potential pathways for the development of these cells. For example, during the germinal center processes, there are poor health from T follicular helper cells during this step and may drive to the differentiation into atypical memory cells, as can we see here. There is another potential pathway that mentioned that the upregulation of inhibitory receptor and reduced BCR signaling may promote the development of atypical memory B cells from classical memory B cells. Another one is that atypical memory B cells could arise during activation of memory B cells in a normal response to infection via an unknown mechanism. And finally, other potential pathway is that atypical memory B cells may represent a population originated from the extrafollicular pathway. So what could be the pathway for the development of atypical memory B cells in response to COVID-19? To address this question, we perform different analyses, including trajectory analysis, starting with transitional B cells, and differentiate into 3 different lineages: switch-activated memory B cells, antibody-secreting cells, and atypical memory B cells, indicators that probably these cells comes from conventional memory B cells through germinal center. Also, we observe that these atypical cells express genes related to germinal centers, and when we run the gene ontology analysis, we observe that the atypical memory B cells express genes related to regulation of B cell receptor signaling pathway, regulation of B cell proliferation, differentiation, B cell activation. So this means that these cells are probably activated and with the BCR signaling. Additionally, we observe that there are some genes and transcription factors related to COVID-19 signature expressed in atypical memory B cells. So if we go back to this slide, probably one of the potential pathway that fits in COVID-19 could be this one, that atypical memory B cells could arise during activation of a conventional memory B cells in a normal response to infection via an unknown mechanism. But in this case, these unknown mechanisms could be the genes and transcription factor related to the COVID-19 response. And as conclusions, we can say that single-cell analysis reveal a high heterogeneity of B cells in response to the different clinical conditions of COVID-19. The different B cell subsets could contribute to the proinflammatory response in several cases. Also, variations in gene usage and somatic hypermutations were observed between atypical and conventional memory B cells, and atypical memory B cells could arise from germinal center during the differentiation of memory B cells and can be influenced by genes and transcription factors characteristic of COVID-19 as a normal response to the infection. I would like to thank our collaborators in Mexico and in Japan, and thank you.
Thank you, Melissa. There are some questions in the chat. Let me see if I can find. For example, what gene markers did you use for the annotation of atypical memory B cells, especially for differential markers compared to activated B cells?
Okay, well, in general, for the conventional, we use CD27, but then we had these clusters of cells that are very similar to memory B cells, but they didn't express enough or high expression of CD27 and CD21, sorry. So we compare these cluster between memory B cells, and we observe this low expression of CD21, CD27, and also expression of other markers such as I don't remember the marker, CXCR5 and CD24 that was previously reported for atypical memory B cells also.
Okay, another question. Oops, it moved. Why the most of the naive activated B cells are mutated and a small portion of them are even highly mutated?
Why the most of the naive activated B cells are mutated and a small portion of them are even... Okay, because even naive B cells, well, naive activated B cells are these cells from extrafollicular response. So even in extrafollicular response, these cells can go through somatic hypermutation at low level, but they can go through this process.
That's another question. Interesting that the T2B cells are present in both critical and moderate, but not the severe group. Are the moderate and severe T2B cells in that cluster the same cell types, or do they differ and happen to collapse into the same cluster in this projection?
Sorry, what is the question again?
It moved. It's from Brad Langhorst.
Okay.
I can repeat again. Are the moderate and severe, or maybe he wants to unmute and ask?
Yeah, I'm happy to. And maybe this is just a dumb question. So if it's confusing, feel free to ignore me. I guess I'm wondering to what extent is that I think it's TSNE or UMAP, I'm not sure which. You have those as one cluster. I couldn't quite tell if it was the same. Like, is there something about those cells that are different between the moderate and the critical group that are, yes, they're in the same cluster here, but is there some property that those cells share that are it's surprising that they skip over the severe, I guess, is the piece that I'm wondering about. Why are those T2 cells not present in the severe grouping? Or is that a false cluster? Is there really two clusters there we're seeing as one?
Sorry, I don't really understand the question.
So this T2 cluster, we go from the light blue to the gold to this sort of red cluster on the right of your TSNE plot. I think that's a TSNE plot.
Yeah.
There's a sort of light blue T1 cells, and then there's these T2 cells. I'm wondering if that cluster of T2 cells is really two clusters. And what's making me wonder that is that I'm surprised that the critical patients are sharing the same cluster with the moderate, but not the severe.
Okay. Well,
in other words, if you clustered it again with a different method, do you think that cluster might separate? And maybe is another way to think about this.
We made the clustering and we identified these three subset T1, T2, and T3 with the level of expression of these canonical markers.
Okay, maybe let's follow up later if this instead of taking time here.
Okay, we can take one more question. Did you see any B cell energy genes in the T3 subset?
No, we didn't observe energetic B cells.
Okay, Melissa, if you have the time, just review the chat because maybe I skipped some of the questions and you can answer. I remember somebody was asking for the specific link to the paper that you are presenting.
I will share the paper through the chat.
Okay, excellent, thank you.